Hi, everybody. I'm Heather Thorstensen, Manager of Communications for Sigma Xi, the Scientific Research Society. Today, for our Meet Your Fellow Companion series, I'm going to be talking with Sigma Xi member Magesh Thiyagarajan. Magesh is an Associate Professor of Engineering at Texas A&M University-Corpus Christi. He is the Director of the university's Plasma Engineering Research Lab, and he leads research involving cold plasma. Magesh, thanks for joining me.
Hi, thank you.
Can you start by explaining what cold plasma is?
Sure. Cold plasma is nothing but plasma in near room temperature. Plasma itself, it's nothing but an ionized form of a gas. We traditionally know the three phases of matter: solid, liquid, and gas, based on how much energy that we feed into it. And when we add more energy to gas, they reach an excited state, and from there it reaches an ionized state. So plasma is a collection of ionized particles such as electrons, ions, and some of the gas particles itself. It's very similar to the blood plasma. That's why the name itself derived from it. It's a mixture of ions and electrons in a cloud of gas, similar to what you would have on a blood plasma, white cells, red blood cells in a plasma medium. So when we make plasmas in room temperature, so the name is called cold plasma.
And one of the projects that you're working on with cold plasma is to fight cancer. Can you talk about how cold plasma is used to fight cancer?
Sure. The working on cold plasmas for cancer research has been one of our recent initiatives. We had been working on cold plasma and seeing their biochemical properties on working with microorganisms and so on. So we also been very aware that the cold plasma produces reactive species. So while exploring the cancer research, we found that some of the cancer cells, they actually require these reactive species for them to go through the self-induced death process. We call it apoptosis. And that process itself require an external supply of an environment with reactive species. And surprisingly, the cold plasmas tend to produce the exact same type of reactive species. So when we explored it, we actually found at a certain amount of plasma exposure or certain amount of reactive species exposure, the cancer cells can actually choose to go from either apoptotic method, and when we have, of course, prolonged exposure or overexposure, the cancer cells go through necrotic process, which is an sudden abruption of cell structure. We could actually begin to see these cancer cells begin to go through this apoptotic or self-induced death process over 24 hours and over 48 hours, which is the actual preferred way for cancer treatment.
And another medical application that you're working on is infections with cold plasma.
Right.
How is infections in cold plasma related?
Um, so the infections can get worse or can be prolonged based on the bacteria can be present on the open wounds, and as well as the tissues that were being damaged over the infection area could take longer time, and it also requires the similar reactive species that body itself generates for healing purpose. So when the cold plasmas, which produces a similar reactive species, if we supply it externally around the wounds, it does two things. One is it sterilizes the area with by killing these any potential bacteria may be present, which could actually further worsen the wound. So when we clear the area with cold plasmas, then the cold plasma supplied reactive species also aids the wound to accelerate the healing process by absorbing the externally supplied reactive species, and it also triggers some of the biochemical reactions for these cells to send signals out for asking for more species for tissue repair. So that project is a very new initiative, and we are still beginning to work on it. And there's a long way to go on wound healing project.
What would be some of the benefits of treating cancer or treating infections with cold plasma rather than treating them with more traditional methods?
That's a very good question. For cancer treatment, it's a new exploratory field. It could be an alternative technology, or it could be used as a supplementary technology, or complementary in addition to the existing technologies. One challenge with existing technologies still today is the severity of the treatment itself, such as the radiation and chemotherapy. It can be very harsh on the patients due to the overexposure or full exposure of the human body. With respect to plasmas, they are less radiation, or in fact it doesn't have radiation for the overexposure of the entire body, for example. So if you have a localized treatment, skin cancer or tumors, you can have it treated very locally instead of inducing them through drugs or radiation therapies. So in a way that it's more localized and it doesn't expose the tissues or the body itself, where you impact lots of healthy cells through the treatment. And with respect to wound healing, it's very similar. Instead of taking a drugs and exposing your entire body to it, and experience more side effects because of the treatment, we are looking at more localized area of treatment, and this is something that you could imagine as a handheld device, or portable enough that you could operate it at home instead of having a treatment done in a hospital setup. So there are scientific as well as the benefits for end users as well using the cold plasma technology.
Can you show us a little bit about what a cancer-fighting cold plasma device might look like? And I know other researchers are working on cold plasma with cancer, so I'd like to know the unique part about your project.
What we develop is cold plasmas in atmospheric air, and we also use some of the noble gases. And the equipment itself that we use, we had developed a number of different cold plasma technologies, but principally based on the cold plasma principle. Some may look like plasma needles, some may look like plasma shower. We have one here that we modified. I could show. It's a probe. It's a probe, and it has, as you could see, it has number of openings from which an array of cold plasma would would uh, would propagate out to the surface, and we usually have it upside down. And when we have the cancer plasmas exposed to it underneath with our cancer cell lines, we treat them for anywhere between a few seconds to couple of minutes for calibrating a number of array of plasma dosages. And our unique part of our research, one second. The unique part of our research is we develop cold plasmas with atmospheric air, so it doesn't require an additional supply of gases as an operating supply. And also we try to make it as portable as possible for other military related applications.
And I wanted to ask you about the portable devices, because I know that that's a project that you have received funding for from the Department of Defense. And I wondered, what is the main goals of having a portable cold plasma device, and what are the special considerations involved in making something like that?
That's a good question. As you said, there's been growing interest on this cold plasma area, and the potential for for wide ranges of biomedical applications, let it be wound healing or infection treatment or sterilization. So there has been a great interest in public as well as in government aspects. Uh, However, traditionally the plasmas or the prototypes or the hardware that requires to make plasmas are traditionally bulky and huge. You would require supply of gases and power supplies, and then the arrangement itself would be usually large in size. So when the military or the Department of Defense was interested in possibility of using such a technology for assisting soldiers or in a distant locations, and there has been several incidents for infections through wounds, or sometimes when they have catheters being used for treatments, and they actually have produce or infect those wounded areas. So there has been a need for distant applications or remote applications for cold plasma for disinfection applications. So military Department of Defense was interested in developing a portable type of plasma source where you could have it in your backpack or something that you can handheld device, or you could have it battery operated in an ambulance setup where you could have it used for initial treatments for disinfection or treatments of that sort. So we developed a range of plasma devices through the support of Department of Defense funding.
And food sterilization is another area that you're working on with cold plasma. Is that very similar to the wound healing where you're using the cold plasma to remove the bacteria?
So far, I talked about the biotechnology aspects of or applications of cold plasma. And I personally myself had worked on nearly a decade on working with cold plasmas for bacterial contamination or decontamination. And we had a literature of different media such as dry surfaces, moist surfaces, and liquids, and so on. And if cold plasma can work on such surfaces, they should be able to work on food surfaces as well. So that's when we started looking at using cold plasmas on food materials. And then we know the food recall is a major issue to our society till today with Salmonella, E. coli, and so on. And when we looked at different produce and poultry, we found variety of bacteria. The results were vary with respect to bacteria or the smoothness of the food structure or surface texture. But overall, we were able to demonstrate the efficacy of cold plasmas on killing bacteria on food surfaces. And we also lately actually tested cold plasma on some seafood because we are located by the Gulf Coast in Texas, and we actually have it tested on certain type of seafood. The principle of operation, it's similar but different. The cold plasma produces the reactive species. They also have the tendency for reacting with bacterial cell surfaces as well as their functioning itself. So once you rupture those cell surfaces as well as their functioning mechanisms, you could actually kill colonies of bacteria in a very short period of time.
What would be some of the benefits of using cold plasma for the food sterilization?
That's a good question. Traditionally, right now, I think the technologies being used are very common type of methods that you would actually use it to clean any surfaces. Right now, depending on different types of poultry or produce, they use water and sometimes warm water or hot water, and in rare cases, they would use some chemicals to decontaminate and then wash it with water. And there has been some radiation technologies being explored, such as gamma radiation and electron beam radiation. So there are issues with the current technologies. Either one is the most traditional heat related or steam related applications that are harder in both in terms of its operation as well as its efficacy, the guaranteed requirement for bacterial reduction. And then the other high energy applications, they are more capital intense and bulky, and there are some risks involved with it. The benefits for cold plasma compared to the existing technologies would be, as I said, it can be made much more user friendly, and it can actually have significant bacterial reduction, provided it's designed appropriately for the specific application type of foods. And then the other benefits for the commercial aspects is it can be made much more cost effective for the amount of treatment one they would choose for compared to the existing technologies.
Can you talk a little bit about where cold plasmas are in terms of regulations and being used in actual society? Are they right now just in labs, or are we using them, or are we going to be seeing them soon out in the general public?
Since I've been working on it, I personally like to see this technology being used in public very soon. And there has been very few commercial interest on this area, because being it's a new field and it's a growing field, and there are very small number of either large companies or small scale companies are looking into taking a product from the lab setup into a commercially viable setup, increasing the technology readiness level to a much higher level. So it could be used by the end user applications. So it's a growing field, and I think this technology will be in the hands of public in a very short period of time, hopefully in a few years. But the interest is there to make it as from the research level into a commercially readily available plasma device that you could buy it off the shelf.
And I know besides the cold plasma, you're also working with laser plasma, and I want to know the difference between the two and what your laser plasma project is.
Cold plasma, as I said, it's a plasma near room temperature. So since it's near room temperature, we use it for a variety of heat-sensitive applications. The laser-induced plasmas that we work on research on, it's mostly for studying the high temperature, high density plasmas that are near to the conditions that exist in the surface of the sun. But in a very microscopic scale. The interest towards laser plasmas, one for the understanding of plasma dynamics with its surrounding media, as being used in the fusion research applications, and also now we are looking at using laser plasmas to produce high temperature microscopic plasmas in liquid medium. In conjunction with materials such as gold or platinum, to produce a very high energy nanoparticles directly into the liquids. So those are some of the areas that we use laser plasmas, which are usually hot plasmas, for either studying the fundamental plasma physics, as well as to study the fundamental aspects of how nanoparticles can be produced directly in liquids. That can be used for a variety of applications as well.
And one of the major focuses of Sigma Xi is to strengthen the health of the research enterprise, and that involves supporting students. And I know that supporting students and outreach is very important to you as well. Can you talk a little bit about what you've been doing for that?
Sure. Outreach is actually part of our culture in our program in our college at Texas A&M Corpus Christi. We reach out to middle school, high school students when they come here for campus tours. We make sure that they visit our plasma lab, and I personally myself provide the demo of some of the plasma equipments, and I also have some of my students to give them and talk to them what is it like working. And we also do some science competitions, and our college actually conducts annual Sigma Psi symposiums that involves students to present papers and conferences. So when we ask our students to assist these type of outreach activities, our students gets really excited, and we form a team, and then we usually use our research team to give us demo for high school students, and as well as our own undergraduate students when they come here for campus visits. We have something called Islander Days, and when we have students ready to apply for engineering programs, we give them a tour of the labs as well. And besides that, being in the minority serving institutions, we have reached to several students in the region where they enjoy more research experiences or hands-on experiences, which usually they don't get it in other conditions. So these are some of the outreach activities that we do. And actually, I personally did a couple of years of engineering competitions where we made students to build catapults and chariots based on basic raw material that we supply. So outreach is part of our culture, and then we feel great about having some of those members joining the STEM education and engineering program, and some of them actually started working in our plasma engineering research lab.
And you have a little plasma device there to show us that maybe you use that during your outreach of what the plasma looks like?
Yeah, so we call this as a plasma ball, and where it produces, again, something similar to a cold plasma. And I'm going to turn it on now. I'm not sure how well you can see it, and there you go. So it puts out a plasma or a rays of plasmas to the outer surface, which is a glass that you see here. And then when you actually have a contact to it, you actually make a lot more denser plasma, and then all the other rays of plasma goes away. And it doesn't burn your hand. If I hold it towards the camera, you could see it gets warmer, but it doesn't burn your finger. And the reason that it makes a contact to your finger, then when you release it, it makes all over the chamber. It's basically because you supply some of the static electricity around your body, or your ground. You act as a ground between one of the terminals and the other terminals. And this plasma, I also use it for to demonstrate during the outreach how this ball can actually light a fluorescent bulb without actually having it electrically connected, basically through electromagnetic radiation being used in this medium, as well as it radiates outside the glass. So when you bring in a flash lamp, it actually lights up based on the electromagnetic radiation comes out of this plasma bulb. So these are some of the tools that we use it to get them started to see what plasma is about.
Well, thank you for showing that, and thank you for explaining your research. It's been great to talk to you.
And it's been a great pleasure talking to you, and I feel glad being a member of Sigma Psi for several years. I'm very happy to share my experiences to the community. So thank you very much.